Thank you so much. First of all, thanks to the organizers of this symposium for inviting me, and thanks to everyone for joining from around the globe. I'll be talking to you today about different enabling tools and algorithms for speeding up synthetic biology-based workflows. We are a software company that has been building a wide range of tools and algorithms that are being used today by different labs interested in running synthetic biology workflows. So my talk today will also be probably more practical than previous talks and focus more on sharing our experience on helping different organizations using these tools and these techniques in some real world settings. So before
Eduardo, can I interrupt just to ask, could you go to full screen mode, please?
Let's see if I could do that. Give me 1 second. Can you still see my screen?
Yeah, I think there is a button called slideshow that puts it on full screen.
Yeah, all right. Does it work?
No, it just looks like it did before, with the preview pane down the left side.
I think it's okay.
If it's not working, let's just carry on in the interest of time. We can quite clearly see the content.
Yeah, all right. Apologies for that. Let me share it again. I think are you still seeing it?
Yeah, we're seeing it as it is. I think in the interest of time, we can just continue as it is.
I'll try to hide maybe the comments and remove the film strip. Maybe you see it slightly better now.
Yeah.
All right. Yeah. So before before I dive more into some of the tools and algorithms, I also want to give you some context about what on our view is driving the need for AI in synthetic biology. So as many of you already know, biological product development is very hard, fragmented, expensive, and importantly, very slow, as has been highlighted this morning by Jim already. And this slowness and difficulty can be explained by a number of factors. It is hard to simulate, what a complex biological system will do, unlike a mechanical system where you can solve Newton's equations, or in an electronic circuit where you can use Maxwell's equations to simulate the system. Complex biological systems are much harder to simulate, and much of the progress that we see being done today rely on gathering a lot of data and of course, training AI algorithms. And in this process of gathering and using biological data, biologists are designing increasingly more complex designs and trying to build very complex libraries of DNA constructs and running and automating high throughput workflows, interfacing with different components, collaborating with colleagues, learning from previous experiences and experiments, and so on and so forth. So the design of a biological product, therefore, has been, and in our view will continue to be in the next several years, a very iterative process that requires being able to efficiently trace the development cycle that goes from designing your biological product or system through the construction of the strains and biological products to characterizing the phenotype of the product in a very systematic way in order to be able to learn from past experiences. And just to give you some numbers from the pharmaceutical industry, only 10% of potential drugs that effectively pass preclinical development and enter the clinical phase are reaching the market. The number of new drugs approved per US dollars spent on R&D has decreased roughly by 50% every 9 years during the past 70 years. The cost of developing and bringing one single drug to the market has almost doubled from what it used to cost $1 billion to over a few $1 billion already during the past decade. And the median time for an emerging company to launch a new drug is now roughly 17 years. So as a result, we've been estimating that roughly a few $1 trillion are lost every year due to inefficiencies in pharmaceutical R&D, and this is mostly because of the slowness in developing a new drug or biological compound. So why can synthetic biology, for example, help in pharma? We see that in the past 10 years or so, R&D in pharma has been shifting from developing small molecules to developing large, complex biologics. And of course, in order to design a biologic, companies and labs need to design and optimize DNA. And a biologic, like an antibody, is significantly larger than a small molecule. A small molecule might contain, say, 25 atoms, while an antibody might contain 3 orders of magnitude more atoms. So this radically expands the dimensionality of the design space that we're working on. And of course, the larger the design space, the more challenging it is to effectively train AI algorithms, and the higher the complexity of the DNA libraries that needs to be designed and built. And this is, of course, as also already been highlighted, not only affecting the pharmaceutical industry, but affecting the entire biotech industry that seeks to use to develop new generation of materials, food, biofuels, enzymes, microorganisms for industrial use, and so on, relying on modern synthetic biology techniques. So what is needed for doing AI in synthetic biology? Firstly, of course, we need to train good models that can be used to optimize a biological product. And where we see an opportunity is not only in developing AI models that can effectively map, for example, from genotype to phenotype, so that you can come up with a better design of a molecule that achieves a certain optimized phenotype, but also an important challenge in industry that is probably less, sexy, if you will, has to do with many times optimizing for the manufacturability of a biological product. That could be, for example, the titer of an antibody produced in a specific cell line. And if, for example, you don't have, say, a few grams per liter of an antibody in your process, then you really don't have a product. Or it might be the titer of a metabolite produced by a microorganism, and that requires doing some sort of metabolic pathway optimization. Or it might be the case that you are scaling up a process, and you need to optimize the operating conditions, for example, of a bioreactor, so that then you can produce that product at an industrial scale. So all these problems are very important. Many times there are no good mechanistic models that can help you all the way through, and that's where AI models can help you solve for manufacturability challenges. Then in terms of the algorithms themselves, we've classified them roughly into 3 different categories: predictive models, generative models, and evolutive models. And of course, the classification of these models is somehow arbitrary, because you can combine all these different types of models in different architectures to create more complex architectures. And in later slides, I'll come back to this and highlight a few examples. Now, as we know, in order to train these models, we need data. And without good tools, standards, and good infrastructure to gather, parse, and manage data sets, training AI models can become quite challenging. Very often, we find data scattered and isolated in places that can make it very hard to find and difficult to use. You will likely need to acquire data from different analytical equipment, bring it together, visualize it, link it to your designs and experimental workflows. and also transform it and parse it to make it ready for downstream analysis, like training machine learning models. So if you're running your own experiments, as I used to do when I was a PhD student, then you might be able to track your own experiments and integrate different data sets in your own laptop. But this becomes particularly challenging when you are collaborating with colleagues in other departments or in other institutions, or if you're using data that is a few years old from a colleague in your institution that just left before you took over his project. It can become quite challenging. So there are a few different tools that have been developed in the past few years. One of them, for example, is EDD, referred as the Experimental Data Depot, which was developed at the Berkeley lab by Hector's group, who is going to be presenting afterwards, and also our own test module, which has been developed in collaboration also with Hector's group, and provides some additional features many times required by large corporations. But the key point here is that if measurements and descriptors in your data sets, such as different genetic elements or operating conditions, are not properly mapped to specific biologically relevant entities and quantities, it will be very hard to integrate different data sets and use them to train AI algorithms downstream. Then in order to get the data, of course, you need to run experiments. And in terms of tools for handling your experimental protocols, you might all be familiarized with a number of tools and electronic lab notebooks and lab information management systems. But in our view, what is enabling the next generation of AI models are larger and more complex data sets coming from executing high throughput workflows that can't be handled by conventional electronic lab notebooks. Complex biological experiments and high throughput workflows are being automated in industry. There are a number of liquid handling technologies, including microfluidic technologies, that are enabling this automation. And in order to run these workflows, you need tools to automatically generate the instructions for these liquid handling robots. And also, very importantly, these tools for running and tracking experimental workflows in synthetic biology usually depend on different materials, including DNA sequences and plasmids and oligos and instructions to run PCR reactions and so on, that need to be, of course, designed. And this leads to the fourth category of tools, design tools needed to running experimental workflows, which are needed for gathering experimental data, which is needed for training AI models, which are ultimately needed for optimizing biology. So there are a number of design tools out there for editing and annotating DNA and protein sequences, or designing a plasmid, or some guide RNAs for editing DNA, or designing simple genetic circuits. But again, where we see an important need is for more advanced tools for designing complex designs, complex combinatorial DNA libraries, hierarchical DNA libraries, and of genetic constructs. And this has been, what has been driving us to, by our industrial partners that are demanding an increasingly level of complexity of the combinatorial libraries that they are designing and building, which already are containing, several thousands of different genetic constructs in a single, DNA assembly reaction. So when designing DNA combinatorial libraries, many times you also want to impose some design rules or constraints, and have the tools that tell you if any of those rules are being violated. Also, in some cases, you might be designing in the DNA space directly, or if you're like designing an antibody, you might be designing the amino acid space, and at some point you might need to reverse translate your amino acid sequences into DNA and call and optimize your sequences. And also that might be critical for optimizing for the manufacturability of the process. So at TeselaGen, building good design tools for synthetic biology has been at the core of what we've been working on the past many years, and our algorithms, facilitate the generation of different DNA assembly instructions for building complex libraries using some of the most modern DNA assembly strategies, including Golden Gate assembly, Gibson assembly, Moplo, user cloning, and so on. Let me spend a few slides highlighting the importance and the need for interoperability. This is something we've been focusing heavily by building an infrastructure that enables interoperability, and has also been a fundamental driver for us. First of all, in our view, we need more tools that can run independently of each other and that can be interconnected with other tools in the cloud. This is typically achieved by connecting the APIs of these tools or services and databases, and we're doing our part and have developed an API for designing, building, and optimizing biology. Also a command line interface or CLI tool, also a public Python API client to help data scientists interface with our API directly from Jupyter notebooks. And we're making these APIs accessible to other users, but we also expect others to do the same and work together on bringing many of these tools together. How can this be done? We're facilitating integration tools that run on what we call integration servers. These integration servers can be configured through a user-friendly interfaces that let you build what we call integration flows, where you can specify how to communicate different APIs, including our own API. This is based on open source technology, originally developed by IBM. And our platform already offers a number of integration flows and integration points in our platform where biologists can connect to external systems. And in our integration flows that connect our platform with different other stakeholders are classified, I would say, roughly in 4 categories. We need integrations with synthetic biology vendors. We need integrations with lab automation equipment. We need integrations with biological databases. And we also need integrations, of course, with other tools and AI algorithms for synthetic biology. So, for example, we've developed an integration flow that communicates TeselaGen with Twist. So if you're designing a complex combinatorial DNA library and you need to design a long DNA fragment, but you're not sure if a company like Twist can synthesize it, you can very easily and quickly use an integration flow that communicates your design tool with a DNA synthesis vendor and check if the molecule is buildable or not, and if not, you might need to tweak your design and speed up the DVTL cycle. We're also developing integration flows that communicate with public databases and registries of parts such as NCBI and JVIs, where you can also easily search for DNA parts, pull them into a design editor, and automatically check, for example, if your parts are in your inventory, check how much it would cost to synthesize a complex library, and so on. So we really think that interoperability and integration tools are really something needed by the industry. So finally, on that front, we've also developed what we call a microservice framework that is in beta, and we're sharing it with different institutions to download it and use it to package their own algorithms and to make their algorithms available with the community. The different machine learning algorithms that we have developed ourselves have already been packaged using this framework. So in a nutshell, in order to do AI for synthetic biology, we believe that it's fundamental not only to develop good AI models, but also make sure that all these different tools needed for designing, building, and testing biological products can be integrated in order to close the so-called DVTL cycle. and also to integrate with a number of relevant tools and algorithms and stakeholders in the synthetic biology community. Now, let me quickly walk you through some applications that we've been working on, either as part of our own internal R&D projects or in collaboration with some other institutions. So let me say that we've been very fortunate to be working with amazing partners during the past several years. I just want to note that unlike my other distinguished colleagues presenting today, we handle proprietary data sets and information that we can't necessarily disclose, or at least I might not be able to get into all the details to protect the confidential IP of our customers. But we do have some collaborations with also with universities and national labs that have led to some work which have been published and that I will be highlighting in the next few slides. So the first example I want to highlight relates to predicting protein expression. We've been testing different deep learning algorithms to predict protein expression in selected model organisms. This is a very relevant and interesting problem in synthetic biology, and our approach was tested on a large protein expression dataset generated by the Arkin lab at Berkeley, where they synthesized 240,000 synthetic sequences that drive the expression of GFP in E. coli. And with this labeled dataset, we've trained and compared different techniques to predict the expression of GFP based on DNA sequence. In the original paper, 8 features were identified that correlate significantly with protein expression. We were also able to find additional features extracted from a latent layer of sequence encoders that, when used in addition to the original features, were able to outperform the predictive power of the existing algorithms. Coming back to one of the questions we had from a previous talk, the challenge here is that most of the public data that we use comes from model organisms such as E. coli and yeast, and we really need more data around other non-model organisms. Another important challenge is, for example, is to predict binding affinities of, say, antibodies with an antigen. We've tested AI models to predict binding affinities of human monoclonal antibodies by training different supervised models using experimental binding affinity data. The challenge here is to work on predictive models have to be trained on very highly dimensional design space, and here it's key to use different transfer learning techniques to train these predictive models on embedding spaces. And also, in many cases, there's a need to do multi-target optimization and maximize, maybe one quantity while minimizing another one simultaneously. Very quickly, we've also been exploring different encoders and decoders to tackle the challenge of dealing with highly dimensional design spaces. We've been, testing different models based on transformer architectures that we've been training with several millions of sequences coming from public databases. Again, like different features have been obtained using this type of models, using the outputs of specific layers of the neural network encoding the embedding, and we've applied these embeddings for predicting then binding affinity of some molecules and improve the performance of the predictive models by doing so. Another important class of AI models that we have been working on are generative models. We've used these techniques to train generative models that learn the probability distribution of certain molecules in a training data set, and then use the model to sample it and generate new molecules or leads that follow the same distribution. We've shown that by using some generative models, we can indeed capture the desired properties of certain peptides. These peptides that we've been using, typically range from 10 to 50 amino acids long. And we've trained these models with data sets of a few thousand known AMPs. and then use the models to generate synthetic libraries of AMPs characterized by having high antimicrobial activity. Probably skip this slide for the sake of time. And finally, also another application of AI for SynBio is for optimizing metabolic pathways. There is a need to many times to optimize the metabolic pathway that produces a specific high-value molecule. The design space here is typically composed of a few key genetic elements, either some variants of an enzyme in the pathway or the promoters driving the expression of that downstream gene coding the enzyme. This problem is typically more, tractable than in previous examples. The design space here might be something between 10,000 to 100,000 different possible designs. And we've used tools based on sequential model-based optimization to optimize for these metabolic pathways. The optimization algorithm, takes into account the expected outcome, but also an associated uncertainty that is used to compute what is known as an acquisition score that can then be used to recommend promising candidates for a metabolic pathway. And there are some trade-offs, of course, when choosing these acquisition functions, and I won't go into detail, but this is also, an application of great interest. And let me end by, highlighting some partners that, have been using some of these tools, including Lancatech, a company that we work very closely with, that have been designing and optimizing microorganisms that eat carbon monoxide and carbon dioxide to produce different biological products. They recently published a paper where, in collaboration with Northwestern University, they report the use of a Clostridium species to break down carbon dioxide and make it into acetone and isopropanol. Very cool application. Showing how a carbon-negative approach can remove carbon dioxide from the atmosphere and produce high-value chemicals. Another collaborator, DTU, has been doing some cool work. Not too long ago, we published a paper together with them and the Berkeley lab where we use AI in combination with mechanistic models to optimize also a metabolic pathway in yeast with the goal of increasing the production of tryptophan, which was achieved by almost twofold after a single DVTL cycle. And just let me finish by highlighting some other academic and partners and government agencies, and we're very happy to explore new partnerships, collaborations with different groups if this is something that any of you might want to discuss. And with that, I would like to thank you for your attention, and thank to all our partners. And if we have a minute, maybe take some questions.